Well, hello, students and YouTubers. Dave Soriano. I'm a chemistry professor with the University of Pittsburgh's Bradford campus in Western Pennsylvania, USA. And since I've gone to the remote education this summer because of the pandemic, I've been recording selected lectures for my students in certain courses. I've been doing modeling work for years, but of course, attention turned to the coronavirus. So I'd like to show you some of the very simple cursory findings that I've come up with regarding the coronavirus's so-called spike protein, which consists of an S1, which docks. It undergoes pretty dramatic conformational changes in binding to the human receptor ACE2 and angiotensin converting enzyme 2. And a lot of intensive research going into this right now. And the S2, it's almost like a rocket with stages. S2 then is involved in transfer of the viral RNA information into the human receptor site after the membranes blend. I'm an organic chemist, but I just want to show you I would call it a very simple approach to trying to get a handle on the features of the S1 Glycoprotein. So next slide I will bring up and get this underway. Now, Wang et al., Professor Wang, Dr. Wang is in the Chinese Academy of Sciences in Beijing, China. That's where you can find him. He published just literally a week ago in Cell 2020, pages 4 to 7, a very, very important paper where he and his colleagues talk about the structural and functional basis of the SARS coronavirus 2, the entry by using the human ACE2 receptor, as I mentioned. And that receptor, incidentally, is basically an exposed helix. That's where the docking phenomenon is taking place. And there is the protein itself, the glycoprotein, is a trimer, identical trimer. And it undergoes, there are two so-called protomers. In organic, we would sometimes call them rotamers. And there's a flap up, flap down, and when it's going to dock to the receptor site, it goes to flap up where the interaction occurs. Now, I don't want to get into the aspects of that in this video, but the paper by Wang is a very important paper because there's X-ray data. Now, what I did was to go to the protein data bank, and I found where Dr. Wang and his colleagues had uploaded an X-ray photograph, and it's Protein Data Bank catalog number 6LZG. And this has been uploaded also fairly recently. And there you can see immediately in the image, the lower orange colored is the portion of the flap that is ready to complex to that exposed helix of the ACE2 receptor site in green. And this is a very, very interesting image that's been uploaded because it focuses on what I want to see right now, the area around that docking region, the flap. And I wanted to get an idea of the profile of it, the surface area. Okay, so what I did was I downloaded that PDB file from the Protein Data Bank, and I imported it into Cygress molecular modeling program platform, which I use in my modeling work. I've published papers in the past where I've done some molecular modeling, aldose reductase, and some other things. And I brought it into Cygress, and then you can see where up in the upper right you have the ACE2 recepting portion. It's an enzyme in a cell membrane, and it's actually a very very exposed. That's part of the problem. The active site is not too far into the interior there, but there's an awful large cavity around that active site. But that's not what's involved here in the docking. It's the S1 flap that is involved in docking to that helix. What I did then, and this is really my point of this video, I did a water rollover. Now, Connolly, years ago, showed that you could roll over a surface of the protein looking for crevices. You could call them craters, because oftentimes drug candidates will bind not to the active site as competitive inhibitors, but remote site inhibitors, inducing conformational changes. Okay, well, I did a surface analysis of what you're seeing is the analysis of the glycoprotein portion, the S1, and I was trying to get a feel for the number of crevices, and one caught my attention, and I don't know if anyone else has seen this. I've circled it in red. The crevice is in blue, and the surrounding area is fully exposed. And remember, I'm using a water 1.4 angstrom rollover to come up with this. And what I want to tell you about this region, this one approach in science for beginning people, undergrads, is this is like man going to the moon, that you do a flyby. We had Kraft circle the moon, get close and study it, looking for landing sites, before you actually then have an approach where you do a lunar landing. So we could use that metaphor to get a handle on this. Now, the next slide is the most important. After identifying crevices, one caught my attention in red. The amino acids in the crevice, aspartate 398, valine 395, tryptophan 353, isoleucine 358, and tyrosine 396. Now, the study that I'm doing here, a very cursory study in the solid state, what's nice is I have one single flap in up form, ready to bond. That's what I'm interested in. So I don't have the other proteins in the trimer to complicate matters. So those are the amino acids in the crevice. Now, interestingly enough, the amino acids in proximity to that designated crevice, arginine 355, arginine 357, lysine 356, tyrosine 489, and asparagine 354. Now, just recently, Skidmore and co-workers in the United Kingdom detected a patch on the S1 glycoprotein that can interact with the heparin family. And heparin has been well used and well established in medical applications. Now, I'm not going to cross over into commenting on that. I'm not qualified. But you can go to this link where you can read the report by Skidmore and co-workers. And what he found was, he demonstrated experimentally that this S1 glycoprotein can interact with members of the heparin family, and it induces conformational changes. And I have a typo there. It's after heparin binding. I'm sorry about that. There's the link that you can go and read the contents of that paper. And if vaccines are not promising, perhaps investigators should consider Skidmore's suggestion that heparin be looked at as a way to induce conformational change in the spike protein, which perhaps would alter docking to the ACE-2. So anyway, just some ideas, and you never know who may watch this video, and it may prompt some ideas for researchers at the forefront. And my job is to instruct my undergraduate students as they prepare for careers in medicine or graduate school where they will be studying under research professors. My job is to prepare students for that aspect of their careers. So I hope that you find some of this very interesting. And thank you so much for watching. Any questions, comments, you can get a hold of me here. Bye for now.